Okay, so let's start with basics. See, all animals, we humans also included, we depend on plants for food, correct? Now tell me one thing. Where do plants get their food from? Think about it. Plants, they make their own food. Simple. This whole process we call it photosynthesis. Basically what happens is it's a physico-chemical process where plants they take light energy and then convert it into organic compounds. That's why we say green plants are autotrophs. Means what? Self-feeders, no? They prepare their own food themselves. Now rest of us, animals, bacteria, even we humans, we depend on plants for food. So what are we called? Heterotrophs. Plants have stomata, basically small openings on leaves, and on the stems they have lenticels. Through these structures, gases can come and go. Oxygen comes in, carbon dioxide goes out. Simple diffusion process, no? The gases just move in and out through these openings. Now think about it. Why plants don't need special organs? First reason, each leaf, each root, each stem part, they take care of their own oxygen. There is no transport of gases from one part to another, understood? Second, plants respire very slowly compared to animals, not as demanding. Third, when photosynthesis happens in green cells, oxygen is actually produced there itself, so no shortage. And fourth, most plant cells are located quite close to the surface, so gases don't have to travel very far. Simple logic, no? Now about the structure. In thick stems, the living cells are arranged in thin layers inside and beneath the bark. The cells in the interior, they are dead. Dead cells, they just provide support, mechanical support only. But here's the key point. The living cells, they are packed loosely with spaces between them. These spaces form a network of air pockets throughout the plant. So oxygen and carbon dioxide, they can diffuse through these air spaces easily. Smart design by nature, no? Basically, each living cell has contact with air, even in thick woody plants. Okay, let me ask you first, why does everything need energy? Think about it. You need energy to eat, to move, to study, to even breathe. Plants, they need energy for absorption, for transport of water and minerals, for growth, for reproduction. Everything alive, they need energy. But where is this energy coming from? We know we eat food, but the energy locked in food, how is it released? How can cells use it? This is what we will understand through respiration. So cellular respiration basically means this. Inside your cells, food molecules are broken down step by step. During this breakdown, energy is released. But here's the clever part. Cells don't just waste this energy as heat. No. They capture it, they trap it, they store it in a molecule called ATP, adenosine triphosphate. This happens in the cytoplasm and in mitochondria. The mitochondria is like the powerhouse, making ATP. Now what exactly is respiration? It is the breaking of carbon-carbon bonds in food molecules through oxidation. When these bonds break, lots and lots of energy is released. The compounds that are being oxidized, we call them respiratory substrates. Usually this is glucose, but proteins and fats can also be oxidized to release energy under certain conditions. Clear? Okay, very important point now. When glucose is oxidized, all that energy is not released suddenly in one big explosion. That would be disaster. No, the cell is smarter. It releases energy in many small steps, like going downstairs step by step. Each step is controlled by enzymes. Each step releases a little energy. This way, the cell can capture this energy without wasting it. So here is the beautiful part. The energy from food oxidation, it is not used directly by cells. No. First, it is captured and stored in ATP. Think of ATP like money in a bank. You earn money, you save it in bank. Then when you need to buy something, you take the money from bank and spend it. Same thing. Cell captures energy from food oxidation, stores it in ATP, and whenever energy is needed for muscle contraction, for nerve impulses, for biosynthesis, for anything, ATP is broken and energy is released. This is why ATP is called energy currency. It is like the universal money of the cell. And one more important thing, after respiration breaks down glucose, what remains? Carbon skeletons. These carbon skeletons, they are not waste. No, cells use them as building blocks to make new molecules, proteins, fats, nucleic acids, everything. So respiration does two things. One, it releases energy for ATP. Two, it provides building blocks for making new cellular components. That is why respiration is so central to life. Now see, if we were to completely burn glucose, like we burn fuel in a furnace or petrol in a car engine, the complete combustion equation is this. One glucose molecule adds 6 oxygen molecules, and what we get? 6 carbon dioxide, 6 water molecules, and huge amount of energy. But here is the problem. Most of this energy is released as heat, not useful heat for making ATP, just wasted as thermal energy. And all this heat in one shot, it would literally cook the cell, destroy it. So what cells do is completely different. Cells follow a strategy. They don't burn glucose in one big step. Instead, they break it down in many small steps. Each step, a little energy released. Each step, an enzyme controlling it. Each step, just right amount of energy to make ATP. Not too much, not too little. This way, maximum energy is captured in ATP bonds and minimum is wasted as heat. This is the cellular strategy for respiration. This is why respiration is step by step. Clear? Okay, now interesting history lesson. When life started on Earth billions of years ago, there was no oxygen in the atmosphere. Can you imagine? Completely oxygen-free environment. And life still evolved. That means organisms developed the ability to get energy without oxygen. Even today, we have organisms that live in oxygen-free environments. Some of them we call facultative anaerobes, means they can manage with oxygen or without oxygen, flexible. Others are obligate anaerobes. They must have anaerobic conditions. They cannot tolerate oxygen at all. So basically, life has ways to extract energy from food without oxygen. This process of partial oxidation of glucose without oxygen, we call it glycolysis. Basically, glucose is broken down to picoleic acid, small molecules, and this happens without oxygen. Glycolysis is so ancient, so fundamental that every single living organism has this pathway. Every bacteria, every yeast, every plant, every animal, everyone can do glycolysis. In anaerobic organisms, glycolysis is the only process for getting energy. In aerobic organisms like us, it is the first step. Very important pathway to understand. So the word glycolysis comes from Greek. Glykos means sugar. Lysis means splitting. So glycolysis means splitting of sugar. Now who discovered this? 3 scientists, Emden, Meyerhoff and Parnas. So it is called the EMP pathway. Very nice history. And remember, in anaerobic organisms, this glycolysis is the only way to get energy, the only respiration process available. So it is ancient, it is fundamental, it is called EMP. Glycolysis happens in the cytoplasm, not in mitochondria. Cytoplasm, and it is present in absolutely every living organism, bacteria, fungi, plants, animals, everyone. Now, in plants, where does glucose come from? It comes from photosynthesis. Photosynthesis makes glucose, sucrose, starch. These are stored. When glycolysis is needed, these storage carbohydrates are broken down, converted to glucose. Now the first step. Glucose comes in, it is phosphorylated, means phosphate group is added to it. The enzyme doing this is called hexokinase. So hexokinase adds phosphate to glucose, making glucose 6-phosphate. This phosphorylated form then changes shape, isomerizes into fructose 6-phosphate. Now both glucose and fructose can go through the same pathway, same subsequent steps. That is how it works. Okay, so glycolysis has 10 reactions, 10 steps. Each step has an enzyme. Each step is controlled. And here's what you need to focus on. Where is ATP used up? Where is ATP made? Where is NADH made? This accounting is very important for understanding the energy balance of glycolysis. Let me walk you through the key steps. First, the ATP usage. Step 1. Glucose is converted to glucose 6-phosphate. This requires 1 ATP. Hexokinase does this reaction. The ATP provides the energy and the phosphate group. Step 2. Fructose 6-phosphate is converted to fructose 1,6-bisphosphate. This also requires 1 ATP. So at the beginning of glycolysis, 2 ATP molecules are invested. The cell spends energy first to prime the glucose. Like investment, no? you invest money to earn more money later. Now, middle of glycolysis, fructose 1,6-bisphosphate is split. It breaks into 2,3-carbon pieces, dihydroxyacetone phosphate, and 3-phosphoglyceraldehyde called PGAL. Now, from this point on, we have two molecules going through the same pathway. It's like one becomes two. And here's the important part. When PGAL is converted to 1,3-bisphosphoglycerate, that is when NADH is made. How? The enzyme removes two hydrogen atoms from PGAL. These hydrogens are transferred to NAD+. NAD+ becomes NADH, and the PGAL itself becomes oxidized. It also gets phosphorylated by inorganic phosphate. So we get BPGA. Beautiful chemistry. Next, ATP synthesis happens. First, when BPGA, that is 1,3-bisphosphoglycerate, is converted to PGA, 3-phosphoglyceric acid, this is an energy-yielding step. The energy released is trapped in ATP formation. One ATP is made. Then, towards the end, when PEP, phosphoenol pyruvate, is converted to pyruvate, another ATP is synthesized. So we get two ATP produced in these energy-yielding steps. This is how glycolysis makes ATP. Okay, so we have broken glucose down to pyruvate. Now, pyruvate is like a crossroads, no? It can go three different ways. The path it takes depends on what the cell needs, what conditions are present. This is crucial for understanding how cells adapt to different situations. The three ways pyruvate can go. 1. Lactic acid fermentation. In this case, pyruvate becomes lactic acid. 2. Alcoholic fermentation. Pyruvate becomes carbon dioxide and ethanol. Yeast does this. 3. Aerobic respiration. Pyruvate enters mitochondria, gets completely oxidized, produces lots of ATP. Which way depends on oxygen availability and organism type. Now, fermentation. In fermentation, glucose is not completely oxidized. Only partial oxidation happens. And it happens without oxygen. So how does it work? Well, pyruvate, which we got from glycolysis, it is converted to either carbon dioxide and ethanol or to lactic acid. Depends on the organism. In yeast, when it does alcoholic fermentation, two enzymes are involved. First, pyruvic acid decarboxylase. This enzyme removes carbon dioxide from pyruvate, leaving acetaldehyde. Then, alcohol dehydrogenase takes this acetaldehyde and converts it to ethanol. This produces alcohol. That's why yeast is used to make beer and wine, no? In other organisms, like some bacteria, the enzyme is different. Lactate dehydrogenase. This enzyme converts pyruvate directly to lactic acid. Now think about your own muscles. When you run very fast, when you do intense exercise, your muscles need lots and lots of energy. Your breathing may not supply enough oxygen fast enough. So your muscle cells, they switch to fermentation. Pyruvate is converted to lactic acid. This is called lactate dehydrogenase doing this. Lactate dehydrogenase reduces pyruvate to lactic acid. And you know the feeling? After intense exercise, your muscles feel sore, burning. That is because of lactic acid accumulation. But this process allows your muscles to keep producing ATP without oxygen. Here's the key point. In fermentation, something very important happens. NADH, that is NADH plus H plus, it gets reoxidized back to NAD plus. Why is this important? Because glycolysis needs NAD plus to keep running. NAD plus is the electron acceptor. When glucose is oxidized in glycolysis, electrons are given to NAD plus, making NADH. But if all the NAD plus is used up and converted to NADH, glycolysis stops. Dead stop. No more ATP can be made. So in fermentation, the NADH is reoxidized. How? By reducing pyruvate or acetaldehyde. The NADH gives electrons to pyruvate, making lactic acid, and NADH becomes NAD plus again. This way, NAD plus is regenerated, glycolysis can continue, ATP keeps being made, even without oxygen. But here's the problem with fermentation. Very little energy is released. Less than 7% of the total energy in glucose is released during fermentation. Compare this to aerobic respiration, which we will see later releases almost all the energy. And not even all of that 7% is captured as ATP. Much of it is lost. So fermentation is not efficient for energy production. It is mainly for survival when oxygen is not available. And the end products? Problem. In alcoholic fermentation, ethanol is produced. Ethanol accumulates in the cell and at some point it reaches toxic levels. Yeast cells actually die when alcohol concentration reaches about 13%. They poison themselves. That is why wine and beer made naturally by fermentation, they typically have about 13% alcohol. No more. If you want higher alcohol content, you have to add alcohol externally after fermentation is done or distill it. In lactic acid fermentation, lactic acid accumulates. You experience this after intense exercise, the burning in your muscles. That burning sensation, that is lactic acid. So fermentation has dangers. Toxic products accumulate. Okay, now aerobic respiration. This is completely different from fermentation. Aerobic means with air with oxygen. So aerobic respiration happens when oxygen is present. And in this process, glucose is completely oxidized. Completely. All the carbon is released as carbon dioxide. All the hydrogen is combined with oxygen to make water. And the energy released, huge amount, captured in lots of ATP. Aerobic respiration is most common in all higher organisms. All of us, we primarily do aerobic respiration, and it happens in the mitochondria, the powerhouse of the cell. So pyruvate, which is made in the cytoplasm from glycolysis, now it must enter the mitochondria. It crosses the mitochondrial membrane and enters the matrix, that is the inner chamber of mitochondria. Once inside, it is ready for the next stage of aerobic respiration. Now, two crucial things happen in aerobic respiration. First, pyruvate is completely oxidized. This means hydrogen atoms are removed step by step. And as carbon is oxidized, 3 carbon dioxide molecules are released. The entire carbon skeleton is broken down. Those electrons from the hydrogen atoms, they are passed to oxygen, and this electron transfer, it is coupled to ATP synthesis. So lots of ATP is made. These two events are linked. Perfect coordination. And here is something important about mitochondrial structure. The matrix is the inner compartment. Krebs cycle happens here. Oxidation of pyruvate happens here. But electron transport, that happens on the inner membrane itself. ATP synthesis also on the inner membrane. This separation, this compartmentalization, it allows for better efficiency and better control. Different things happen in different places. Very organized, very efficient. Okay, so pyruvate enters the matrix. Now what happens? It undergoes oxidative decarboxylation. What does that mean? Oxidation means electrons are removed. Decarboxylation means carbon dioxide is removed. Both things happen. And this is done by a complex enzyme called pyruvate dehydrogenase. It is not one enzyme, but a group of enzymes working together, a multiprotein complex. Now, what does pyruvate dehydrogenase need to do its job? It needs coenzymes. First, NAD+. This accepts the electrons that are removed during oxidation. It becomes reduced to NADH. Second, coenzyme A. This is a special carrier molecule. It accepts the acetyl group that remains after carbon dioxide is released. Third, magnesium ion Mg2+. This is a cofactor. It helps the enzyme work. So 3 things. NAD+, coenzyme A, Mg2+. All needed for the reaction. So the equation, pyruvate plus coenzyme A plus NAD plus Mg2 plus and pyruvate dehydrogenase catalyzes this. And we get acetyl coenzyme A plus carbon dioxide plus NADH plus H plus. One pyruvate produces one acetyl-CoA, one carbon dioxide and one NADH. Since one glucose makes 2 pyruvates, oxidation of both pyruvates gives 2 acetyl-CoA and 2 NADH. This is the link between glycolysis and Krebs cycle. Very important link. Remember, one glucose gives 2 pyruvates. Each pyruvate gets oxidized by pyruvate dehydrogenase. Each oxidation produces one NADH. So from one glucose, 2 NADH molecules are produced at this stage. These 2 NADH will be very important later because they will donate their electrons to make ATP. But first, the glucose carbons go into acetyl-CoA and enter the Krebs cycle. So acetyl-CoA is the key molecule. It contains an acetyl group that is 2 carbons attached to coenzyme A. This acetyl-CoA is like a loaded truck ready to deliver its cargo to the Krebs cycle. The acetyl group will be oxidized and the carbons will be released as carbon dioxide. The electrons removed will make NADH and Fa-2 for the electron transport system. Now, acetyl-CoA enters the Krebs cycle. Krebs cycle is also called tricarboxylic acid cycle or TCA cycle, named after the scientist Hans Krebs, who discovered and mapped out this cycle. This cycle is the heart of aerobic respiration. In this cycle, the acetyl group from acetyl-CoA is completely oxidized. The carbons are released as carbon dioxide. The electrons are captured in NADH and FADH2. These reducing molecules then go to the electron transport system to make lots of ATP. That is the flow. Okay, so the Krebs cycle starts. Acetyl-CoA comes in with its 2-carbon acetyl group. It meets a 4-carbon molecule called oxaloacetic acid, OAA. The enzyme citrate synthase catalyzes their joining. They condense together and water adds. What do we get? Citric acid, a 6-carbon molecule. 6 carbon because 4 plus 2 equals 6. And coenzyme A is released as a product. Now we have citric acid. This is the first molecule of the cycle. From this point on, the cycle turns. Next, citrate rearranges. Isomerization happens. Oconittase enzyme does this. Citrate becomes isocitrate. Still 6 carbons, but the structure is rearranged. This rearrangement is important because it positions the molecule correctly for the oxidations that follow. Think of it like arranging blocks so they fall in the right direction when you push them. Now the cycle continues with oxidations. First, decarboxylation. Isocitrate loses 1 carbon dioxide. What remains? Alpha-ketoglutaric acid, 5-carbon compound. During this decarboxylation, electrons are removed and captured by NAD+, forming NADH. Then second decarboxylation, alpha-ketoglutaric acid loses another carbon dioxide. What forms? Succinyl prototype. The coenzyme A is attached. Again, NAD+ captures electrons and forms NADH. So we have removed 2 carbons as carbon dioxide, captured 2 NADH. The acetyl group is being oxidized step by step. Beautiful. Now, very important step. When succinyl-CoA is converted to succinic acid, energy is captured directly as ATP. This is called substrate level phosphorylation. Here is what happens. GTP, guanosine triphosphate, is first synthesized. This happens directly from the energy of the substrate oxidation. Then GTP is converted to GDP and at the same time ATP is synthesized from ADP. So we get ATP directly. This is different from electron transport phosphorylation, which we will see later. Here it is direct substrate level. ATP made directly from the energy of breaking bonds. Only 1 ATP per cycle, but important. Now FADH2 formation. During the conversion of succinic acid to fumaric acid, electrons are removed and captured by FAD. FAD becomes reduced to FADH2. This happens at one specific point in the cycle. FADH2, like NADH, will later donate its electrons to the electron transport system to make ATP. And NADH formation, 3 places in the cycle where NAD+ is reduced to NADH. So you have multiple NADH molecules generated, each one an electron carrier, each one ready to donate electrons to electron transport and make ATP. So from one turn of the Krebs cycle, we get 3 NADH, 1 FADH2 and 1 ATP directly. Now here is crucial for the cycle to keep turning. What is needed? First, oxaloacetic acid must always be available. Without it, there is nothing for acetyl-CoA to condense with. Second, NAD plus and FAD must be regenerated from NADH and FADH2. If NAD plus gets used up, if the cycle makes NADH but there is no way to make NAD plus again, then the cycle stops. It cannot continue. So these are the requirements. This is why the electron transport system is so important. It regenerates NAD plus and FAD by accepting electrons from NADH and FADH2. Okay, so this is the summary equation for one complete turn of the Krebs cycle. One pyruvate goes in. It needs 4 NAD plus, 1 FAD, 2 water molecules, 1 ADP and 1 phosphate. And what comes out? 3 carbon dioxide molecules, complete oxidation, 4 NADH, 1 FADH2, 1 ATP, all happening in the mitochondrial matrix. This equation shows the net result of the entire cycle. carbon completely oxidized, energy captured in NADH, FADH2 and ITP. This is very comprehensive. But remember, one glucose produces two pyruvates, so everything doubles. So what does this cycle accomplish? Complete oxidation of pyruvate, all three carbons become carbon dioxide. Lots of electron carriers, NADH and FADH2H, are the real treasures of this cycle. These will go to electron transport and make lots of ATP and one ATP directly from substrate level phosphorylation. The real value of Krebs cycle is these electron carriers. They hold the energy that will be used to make many more ATP. This is the clever design of aerobic respiration. So by now, glucose has been completely broken down. All the carbons are gone, converted to carbon dioxide. How? Glycolysis breaks glucose into two pyruvates. Then each pyruvate is oxidized, losing one carbon as carbon dioxide. Then Krebs cycle turns twice, once for each pyruvate, and removes two more carbons per turn as carbon dioxide. So all 6 carbons from glucose are now carbon dioxide. Complete oxidation finished. Now what about energy? How many electron carriers have been made? From glycolysis to NADH. From oxidative decarboxylation of 2 pyruvates, 2 more NADH. From 2 turns of Krebs cycle, 1 turn for each pyruvate, 4 more NADH. Total 8 NADH. Plus, from 2 turns of Krebs cycle, 2 FADH2. These are electron carriers. They hold the energy that has been extracted from glucose. 8 NADH plus 2 FADH2. These are the treasures. Direct ATP synthesis. 4 molecules. 2 from glycolysis, 1 from each turn of Krebs cycle. Only 4. When I started talking about aerobic respiration, I said lots of ATP will be made. But so far, only 4. You might be wondering, where is the promised energy? Why all this talk about ATP if only 4 are made? The answer is, in those 8 NADH and 2 FADH2. These molecules hold the energy. They have not yet released it. They are loaded, ready to donate electrons to electron transport. And when they do, that is when the massive ATP production happens. That is why we need electron transport system. That is why we need oxygen. Okay, now we come to the electron transport system. This is where the action is. This is where the big energy payoff happens. The ETS is a pathway embedded in the inner mitochondrial membrane. Think of it like a chain. Electrons pass from one link to the next, from one carrier to another. Each step is controlled. Each step is precise. This is located specifically on the inner membrane of the mitochondria. What does the ETS do? It takes the NADH and FADH2 molecules that were loaded with electrons from glucose oxidation. It removes those electrons. It passes those electrons through a chain of carriers. Each carrier passes the electron to the next carrier at a lower energy level. Eventually, at the end, the electrons reach oxygen. Oxygen accepts them and becomes reduced to water. Simple flow. Electrons travel down the chain, reach oxygen, combine with water. But during this journey, something very important happens. Energy is captured. So the ETS starts with complex 1, NADH dehydrogenase. NADH from the matrix comes to complex 1. Complex 1 removes electrons from NADH. These electrons are passed to ubiquinone. Now NADH becomes oxidized, converted back to NAD+. And ubiquinone, having received electrons, is now reduced ubiquinone. This is the first step of electron transfer in the chain. Now ubiquinone, it is a molecule that is soluble in the lipid membrane. It sits in the inner membrane. It can receive electrons from complex 1 from NADH, but also it can receive electrons from complex 2. Complex 2 passes electrons from FADH2 to ubiquinone. So ubiquinone is a central hub. It collects electrons from multiple sources. When it has electrons, it becomes ubiquinol, the reduced form. Then ubiquinol passes these electrons to the next complex, complex 3. Nice relay. Now complex 2, this is where FADH2 enters the electron transport chain. FADH2 is oxidized and its electrons are passed to ubiquiron. Complex 2 is the entry point for FADH2 electrons. So we have 2 entry points. Complex 1 for NADH, complex 2 for FADH2. Both feed into ubiquiron. Both contribute electrons to the chain. Now complex 3, cychrome BC1 complex. Ubiquinol reduced to ubiquinol brings electrons to complex 3. Inside complex 3 are iron sulfur clusters and other electron carriers. Complex 3 processes these electrons and passes them to the next carrier, cychrome C. Cychrome C is ready to receive electrons and carry them to the next complex. Cychrome C is interesting. It is a small protein. It is not firmly attached to the membrane. It can move. It shuttles between complex 3 and complex 4 carrying electrons. Think of it like a delivery person, running back and forth, carrying packages of electrons from one complex to the next. This mobility allows efficient electron transfer. Finally, complex 4, cychromes oxidase. This is the end of the line. Electrons reach here from cychrome C. Complex 4 has cychromes A and a 3 and copper centers. Here at complex 4, the electrons from cychrome C are finally passed to oxygen. Oxygen accepts these electrons. Oxygen gets reduced. Electrons combine with oxygen and hydrogen ions to form water, H2O. This is where oxygen finally comes in. This is why oxygen is essential. This is the terminal electron acceptor step. Now, as electrons move through the chain, energy is released. This energy is captured by complex 5, which is ATP synthase. ATP synthase uses this energy to synthesize ATP from ADP, an inorganic phosphate. So ATP is made right here in the electron transport chain. The energy released during electron transfer is converted to ATP. Beautiful coupling. Now the key difference, when one NADH donates its electrons, how many ATP are made? 3 molecules. Why 3? Because NADH enters at complex 1, which is at the beginning of the chain. Electrons travel through many steps, releasing energy at each step. That energy drives ATP synthesis at 3 points. In contrast, when one FADH2 donates electrons, how many ATP? 2 molecules. Why only 2? Because FADH2 enters at complex 2, which is later in the chain. Electrons travel through fewer steps, less energy released, less ATP made. So NADH is more valuable than FADH2 because more ATP is made per NADH. Now, oxygen. Where is oxygen used? In respiration, only at the very end. At complex 4, oxygen does not participate in Krebs cycle. It does not participate in glycolysis. It does not participate in complexes 1, 2 or 3. Only at the end, at complex 4, oxygen accepts electrons. Oxygen combines with hydrogen ions to form water. That is it. That is the only chemical role of oxygen. But it is vital. Why is oxygen so vital if it only participates at the end? Because without oxygen accepting electrons, the entire system stops. Think of oxygen like a drain. Without a drain, the sink fills up and nothing else can flow. Similarly, without oxygen accepting electrons at the end, electrons back up in the chain. No more electrons can move. The whole chain stops. No electron movement, no proton pumping, no ATP synthesis. The organism cannot survive. This is why aerobic organisms absolutely need oxygen. Not because oxygen participates in many reactions, but because oxygen is the final outlet. Without it, nothing flows. This process of making ATP coupled to electron transport is called oxidative phosphorylation. Why oxidative? Because oxidation-reduction reactions are happening. Oxidation means electrons are removed. Reduction means electrons are added. These redox reactions release energy that drives ATP synthesis. Phosphorylation means making ATP by adding phosphate group to ADP. Oxidative phosphorylation. The term tells you what is happening. Oxidation-reduction reactions driving phosphorylation. Unlike photophosphorylation, where light energy drives the process, here redox energy drives the process. Now ATP synthase. This is the actual machine that makes ATP. It is made of 2 parts, F1 and F0. F as in the letter F. F1 and F0, like a molecular engine. And this engine sits on the inner mitochondrial membrane, bridging the matrix and the intermembrane space. The F1 part is a big protein complex that sticks out into the matrix. This is where ATP is actually made. Inside F1 are catalytic sites. These sites grab ADP and phosphate and join them together. Chemical reaction happens. Bang! A TP is formed. high energy bond is created. F1 is the factory. The F0 part is embedded in the membrane. It forms a channel, a tunnel, through which protons can pass. Only protons allowed, other ions? No. Very selective channel. And here is the clever part. As protons flow through this F0 channel, the mechanical movement of the channel couples to F1. It causes F1 to rotate. This rotation drives the catalytic action. Proton flow drives rotation. Rotation drives ATP synthesis. Mechanical coupling. How many protons must flow to make 1 ATP? 4. 4 protons must pass through F0 from the intermembrane space into the matrix for each 1 ATP made. These protons flow down the gradient from high concentration outside to low concentration inside. This downhill flow is the energy. Electron transport complexes pump protons out, making high concentration in the intermembrane space. Then protons flow back in through F0, and this flow drives ATP synthase. Very efficient, very elegant. Okay, so to calculate ATP yield, we must assume certain things. First assumption, pathways happen in order. Glycolysis first, then oxidative decarboxylation, then Krebs cycle, then electron transport, one after another. Nice and sequential, like a factory assembly line. Second assumption, all NADH made in glycolysis, it enters the mitochondria, it gets oxidized in the electron transport system. No NADH is wasted. All of it contributes to ATP synthesis. 3 ATP per NADH. Third assumption, no intermediate is withdrawn from the pathway. All pyruvate goes to Krebs cycle. All acetyl-CoA goes through Krebs cycle. Nothing is diverted for making amino acids, fats, or anything else. Complete oxidation. Nothing else happens. Fourth assumption, only glucose is being oxidized. No proteins, no fats, no other carbohydrates, just glucose. Pure glucose respiration. Nothing else interfering. But in real cells, completely different. All pathways are happening at once. Glycolysis, Krebs cycle, electron transport, all running simultaneously, not taking turns, all together. And there is feedback. If Krebs cycle is full, glycolysis slows down. If ATP is high, everything slows down. Constant regulation, very dynamic. And intermediates, they are constantly being withdrawn. Pyruvate is pulled out to make amino acids. Acetyl-CoA is pulled out to make fats. Krebs cycle intermediates are pulled out to make other things. The pathway is not a closed loop. Things are going in and out constantly. And ATP, it is used immediately. The moment it is made, it is used. Cell does not wait to accumulate ATP at the end. Enzymes are not just running at fixed speeds. They are controlled. Allosteric regulation molecule binds and changes enzyme shape. Covalent modification, phosphorylation adds or removes groups. Cofactor availability. If NAD plus is low, the pathway slows. Hormones, they signal the cell. Multiple control points, so enzyme rates vary. Some fast, some slow, depending on what the cell needs. So the calculation of 38 ATP per glucose, that is the theoretical maximum. It is an approximation. In a real cell, the actual ATP produced might be different. More, less, depends on conditions. But the exercise is still valuable. It shows how efficient aerobic respiration is, how much energy is captured, how clever the cell is. Compared to fermentation, which gives only 2 ATP, aerobic respiration is super efficient. That is why aerobic organisms can do so much more with their energy. That is why we can have big brains and muscles. Fermentation could not support that. Okay, let us compare these two processes. First point, breakdown extent. In fermentation, glucose goes to pyruvate, then stops. Pyruvate becomes lactic acid or ethanol. Only partial oxidation. Carbons not completely broken down. In aerobic respiration, glucose goes all the way. Every carbon becomes carbon dioxide. Every hydrogen becomes water. Complete oxidation. Big difference in completeness. Second point, ATP produced. Fermentation, 2 ATP. That is it. One from each glycolysis cycle. Remember, glycolysis made 4 but used 2. Net gain 2. In aerobic respiration, 30 to 38 ATP. Huge difference. 15 times more ATP from aerobic. No wonder aerobic organisms can do so much more energy available. Third point, NAD plus regeneration speed. In fermentation, NAD plus is regenerated slowly. The reduction of pyruvate takes time. This slows down glycolysis. The glycolytic rate is limited by NAD plus availability. In aerobic respiration, NAD plus regeneration is vigorous. Rapid, the electron transport system very quickly oxidizes NADH, regenerating NAD plus. This allows glycolysis to go fast. Much more glucose can be oxidized per unit time. More ATP production per unit time. Much more efficient, much more rapid. Okay, so traditionally, respiration has been called a catabolic process. Catabolism means breakdown. Break down glucose, break down fats, break down proteins, release energy, make ATP, all breakdown. That is the traditional view. But now the real view, respiration is amphibolic. Amphibolic means it does both breakdown and synthesis, both catabolism and anabolism. This is the correct understanding. Example, fatty acids. When you eat a fatty acid, what happens? It is broken down to acetyl-CoA. Acetyl-CoA enters Krebs cycle, oxidized, energy extracted. But when your cell needs to make new fats, acetyl-CoA is withdrawn from the respiratory pathway, not oxidized. Instead, used as building block to make new fatty acids. Same acetyl-CoA, sometimes used for energy extraction, sometimes used for building. Amphibolic. Another example, proteins. When you eat protein, what happens? It is broken down into amino acids. Amino acids are deaminated, amino group removed. Carbon skeleton enters respiration. Could be pyruvate, could be acetyl-CoA, could be Krebs intermediate. These are oxidized, energy extracted. But when your body needs to make new proteins, respiratory intermediates are pulled out. Oxaloacetate from Krebs is used to make amino acids. Alpha-ketoglutarate acid used to make glutamate. Pyruvate used to make alanine. Intermediates are diverted from oxidation towards synthesis. Amphibolic. Okay, so glucose is the favorite, the preferred substrate. It enters glycolysis right at the start. 6 carbons, perfect for the pathway. Most ATP extracted, but that does not mean it is the only substrate. What about other carbohydrates? Sucrose, starch, other sugars? They are first converted to glucose. Sucrose is split into glucose and fructose. Starch is broken into glucose units. Then these monosaccharides enter glycolysis. So all carbohydrates eventually become glucose and enter the pathway. What about fats? Triglycerides are broken into glycerol and fatty acids. Fatty acids are oxidized through beta oxidation, producing acetyl-CoA. Acetyl-CoA goes directly to Krebs cycle. It skipped the early part of the pathway. So fats do not generate as much ATP per carbon, but they still produce ATP. Glycerol, the other product of fat breakdown, is converted to PGR and enters glycolysis. So fats are partially processed, partially bypass early steps. What about proteins? Proteins are broken down by proteases into amino acids. Each amino acid is deaminated. The amino group is removed and converted to urea. The carbon skeleton then enters the pathway, where depends on the amino acid structure. Some enter as pyruvate, some as acetyl-CoA, some as Krebs intermediates like alpha-ketoglutarate or oxaloacetate. All different entry points, all contributing to energy production. Now, a new concept, respiratory quotient or RQ. What is it? It is a ratio, a simple ratio. Carbon dioxide that comes out divided by oxygen that goes in. RQ equals CO2 divided by O2. That is it. Simple math, but very informative. Now here is the key. RQ value depends on what substrate is being oxidized. Different substrates produce different RQ values. Why? Because they have different ratios of elements. Carbohydrates, fats, proteins, they have different compositions. When oxidized, they produce different ratios of carbon dioxide to oxygen. By measuring RQ, you can tell what substrate the organism is using. So RQ is like a diagnostic tool. You can measure RQ of an organism, and from that number you know what it is metabolizing. Rest period, 1 RQ value. Exercise period, different value. Different food eaten, different value. Very clever use of simple chemistry. shows what is happening metabolically inside the organism without needing to measure directly. So when carbohydrates are oxidized, what is the RQ? 1. Exactly 1. For every oxygen that goes in, 1 carbon dioxide comes out. Why 1? Chemical reason. Carbohydrates have a special composition. They contain carbon with water attached. When oxidized, the math works out perfectly to give 1-to-1 ratio. For example, glucose C6H12O6 plus 6 O2 gives 6 CO2 plus 6 H2O. So 6 carbon dioxide molecules out, 6 oxygen molecules in, RQ equals 6 divided by 6 equals 1. Perfect one. Simple. So if an organism has RQ of 1, what is it doing? Oxidizing carbohydrates. That is all. Glucose, sucrose, starch, any carbohydrate all give RQ of 1 when completely oxidized. So RQ1 means carbohydrate metabolism. Very useful information. Now fats. When fats are oxidized, what is the RQ? Less than 1, 0.7. 0.8, less than 1, not equal to 1 like carbohydrates. Why? Chemistry again. Fats have less oxygen in them compared to carbohydrates. So more oxygen is needed from outside to oxidize them. More oxygen goes in, but not proportionally more carbon dioxide comes out. Hence, RQ less than 1. Exam activity man say 12 cal per liter. A fat formula is C51H98O6, a big molecule. When you oxidize 2 molecules of tripalmitin with 145 molecules of oxygen, what do you get? 102 molecules of carbon dioxide plus 98 water molecules. Let's calculate RQ. RQ calculation 102 divided by 145. That equals 0.7. 0.7. This is the RQ for fats. Always less than 1, always around 0.7 to 0.8. Because fats need more oxygen relative to the carbon dioxide produced. Now proteins. When proteins are oxidized, RQ is approximately 0.9 between 1 and 0.7. Why? Proteins contain nitrogen, carbon, hydrogen, oxygen and nitrogen. So oxidation is more complex. Deamination has to happen. Nitrogen is removed. The math gives RQ of about 0.9 between fats and carbohydrates. Now important reality. In living cells, never pure substrate, never just glucose, never just fat, never just protein, always mixture. All three are being oxidized at the same time, just in different proportions depending on circumstances. When do RQ values vary? After you eat carbohydrates, RQ is close to 1. Body is oxidizing lots of carbs. During fasting, hours without food, RQ drops to 0.7 or even lower. Body is oxidizing fats. During exercise, more proteins oxidized, RQ changes too. So measured RQ in a living organism, it is somewhere between 0.7 and 1.2 depending on what the body is doing. The RQ value tells you the metabolic state. Very useful information from one simple measurement.
Meet Iyal, your AI study companion. Transform PDFs into smart study guides, practice tests and flashcards for UPSC, NEET, banking and any competitive exam.
Master your exam preparation with Iyal.